my dear little champions, I hope that you are doing as wonderful as you are, and I welcome you all to PU2 Brahmastra Series. Today is day 3 and today we are going to talk about biology. The chapter that I took is Principles of Inheritance and Variations. Very important chapter because it will give you 13 marks, which is not a joke. And now we will see, according to the blueprint given, what type of questions can you expect from this chapter and how many of them? Okay, so 1 marker question, there will be 1 question. It can be an MCQ or a fill in the blanks, but with the model question paper that I saw, it was always an MCQ that is asked from this particular chapter. Okay, now for 2 marker question, there will be 1 such question over there in your question paper. 1 2 marker question. Okay, then there will be 2 5 marker questions. So here only you will get 10 marks out of this. So 10, okay, you will get 10 marks, then 2 marks over here, then 1 mark over there, makes a total of 13 marks, which is not a joke, as I said. Okay, and what would I be discussing in today's session? We'll be talking about 10 MCQs, we'll be solving 2 marker questions, almost 10 of them, and then 5 marker question, 5 of them, which is a good deal. So with this, I'm telling you, you are preparing yourself for this chapter. Any questions that would be coming from here itself or would have resemblance to the questions that I am discussing right now. Pretty sure about that. Okay, so it is being made depending on the previous year questions, how many of them were repeated, what were the concepts that were usually given for making questions and stuff like that. So please don't be worried, just sit back and just listen to me carefully. Okay, so let's start off with the first MCQ that we are having over here. 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color normal body and white eye color miniature body is due to. So the question says there is 37.2% recombinant of what? Drosophila. That means new combination, new type of Drosophila that you are finding when T.H. Morgan did his experiment. Okay, why is this much recombination there? What is the reason behind it? That is the question. So what do you exactly mean by recombination? For that you have to understand this. Suppose there are 2 chromosomes. Okay, in one chromosome here you have capital B, here you have small b gene, and here you have capital C, and here you have small c gene. Okay, so I am coloring it in yellow and here I will be coloring it in green. So what is the thing between chromosomes is that they will undergo linkage. I mean, they basically exchange their materials. Okay, they exchange their material. For example, this guy could come this side and this thing could come this side, making it a new combination of sets of genes. Okay, so for example, after the recombination has taken place, it will look something like this, where the capital C have come over here. This is capital C, my dear children, and the small c will be somewhere around here. So it has got exchanged. This is what you mean by recombination that is being taken place. So if you see, the initial combination was like this, but now you have got a new combination of genes. Well, this time initially if the chromosome had to go out to get into a gamete, you would get a combination like this, but now you are getting a whole different combination where capital B and capital C is there. You get it? This is what you mean by recombination. Now recombination, if I say 37.2% of recombination has happened, that means it's a very big deal. And how can you expect this big amount of recombination to happen, my dear children? It will happen only when the genes are located at a distance. Suppose the situation was like this, where the capital B is not here and the small b is also not here. Instead, it is present somewhere here, very close to this particular gene. Now, whenever recombination will happen, whatever you do, okay, the new thing that you could be finding would be what? It would look exactly like this, where you would be having small b, capital C over here and capital B, small c over here. It's the same combination that you found, just that they just interchange their position, that's all. So basically, what do you understand from this? That is, if the genes are not linked closely, they will show more amount of recombination. So this is what you mean by linkage. If I say that they are not closely linked, they are far from each other. Okay, not closely linked, that means they are far. That means they will show more recombination. Okay, now on the other hand, if they are close to each other, If they are close to each other, will they show much recombination, my dear children? Do you see no combination, new combination being formed? No. You will see less recombination. Now you have to choose from this. So loosely linked, so here you see more amount of recombination. So you have to answer according to that. Loosely linked and shorter distance between the genes. Is it shorter distance between the genes that recombination will happen? No. Tightly linked and shorter distance between the genes? No, children. If they are tightly linked, they won't be showing recombination. So loosely linked and longer distance between genes? Yes, they are loosely linked, as in they are not close to each other and they have longer distance. Every time it is loosely linked, that means they will be having longer distance as well. And this leads to what? Higher amount of recombination, which is shown over here. Usually it comes out to maximum amount of recombination is 50% recombination. Okay, so basically over here 37.2 is a very big deal. Okay, 1%, 1.7% is a small deal. So here you see loosely linked and longer distances between the genes, which is why they easily show what? Recombination. Got it? This is a question from your model question paper that was given this year. Okay, cool, works. So let's go ahead with the next question that we are having. In her dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes what? So when he did a dihybrid cross, okay, when he took two character, for example, I'll take round and yellow seeds, okay, round yellow and then wrinkled green. When you are taking two character and you are making hybrids out of it, you call this a dihybrid cross, new hybrids, okay. So basically when you cross it, you at the end of F1 generation, you cross it first, then you will get a combination of plants, then again you will cross this, you get F2 generation, in which the ratio is something like this, where 9 is basically what? The parental type, that is round yellow. The parental dominant is 9. 1 will be the parental recessive character, that is wrinkled and green. And these guys will be the new combination, which I didn't see in parents, okay. So it will be, round was always with yellow, but this time we are going to make round with what? Green. So this is a new combination that I found. Over here, there is one more combination, new combination, that is wrinkled, which was always with green, this time it will be with yellow. So you were able to see this. Why? Why were you able to see this? Because children, the alleles which are there, which are responsible for all of these things, okay, they are going to independently assort from each other. For example, if I am saying that this is capital R, this is small r, now if I say this is capital Y, this is small y. Okay, so I cannot think that, for example, this is a plant and plant is having these chromosomes. I cannot assume that, okay, capital R, this gene, sorry, this particular chromosome will always go with this particular chromosome in the gametes. Obviously, gametes had to be formed, and in gametes, what is happening, children? From one pair of allele, only one would be going. So either this will be going or this will be going. So suppose I took R, capital R from here. From here, I cannot say that I will always take capital Y. I can take small y also. So in a gamete, I can have this chromosome and this chromosome. In the next gamete, I can have this chromosome and this chromosome. This is known as law of independent assortment. So they are basically segregating independently. They are not dependent upon each other. They are not saying I will only go with capital Y. It's not like that. Okay, which is why you see new combinations. Okay, if they always went together, you never would have seen newer combination. You see new combination because they are independently assorting, my dear children. So, alleles of two genes are interacting with each other? No. It's a polygenic inheritance? No. It is a multiple allelic inheritance? No. The alleles of two genes are segregating independently. They are moving. For the two genes that you are having, for the shape as well as for the color, both of them are segregating independently, which is why you are able to see this particular ratio. This is again a question from the model question paper 2024-2025. Works? Now, the next question that we are having. A pedigree chart is given below. Identify the trait responsible for this inheritance pattern. So, over here, this is shaded, this is shaded. I hope you know what do you mean by the structures? So this stands for male, okay, this stands for female. Fine. So when they are shaded, when it is totally colored, that means this particular male is diseased, has some disease. When it is not colored, that means they are fine, they are fine. So as of now, they are fine only, they do not have any problem. Over here you see diseased male is there, diseased female is there. Again you see diseased male. So now you are asked, this pedigree chart shows what type of inheritance. What is this particular disease that is being carried around? Autosomal dominant trait. What do you mean by autosomal dominant trait? Any idea? What do you mean by autosomal dominant trait? Basically, I have to make you understand one thing. So we human beings, we have 23 pair of chromosomes, okay? We have 23 pair of chromosomes. Now out of this 23, no, the last 22nd, okay, 22 of them, basically they are known as autosomes. They do not have anything to do with the sex of the organism. They are just there, responsible for making our proteins in our body, giving us our physical characters and everything. The 23rd one, the 23rd pair is the sex chromosome pair. Okay, and it is known as allosome. It is known as allosomes. These are known as autosomes. So whenever you see some autosome dominant trait, or basically, so you are talking about autosomal chromosome, something is happening over here in them. So autosomal dominant trait. So if it is dominant, then basically first thing, if it is dominant, then both the parents would have had this thing, the disease. Because in dominant, for example, A and A is there. If it is dominant, then obviously this particular organism who is having this will have this disease. And suppose the parents do not have this. Suppose the parents are not having any disease. That means that is not dominant. As simple as that. So autosomal dominant trait is removed. Autosomal recessive trait, that could be possible, children. Recessive trait is something that will be shown up when you have both the genes like this. When both are recessive, then only it will be shown. So there are chances that, okay, here you had a condition like this, but now when it's fused, sometimes at the end you might have got something like this, which might have showed up the disease. Similarly, here also you might have got that thing, which is why you got the disease. Now I will say it is not X-linked, it is not X-linked dominant or X-linked recessive. Why, children? Here if I see, basically there is nothing to do with the sex of the organism. There are so many children being produced and only 2 of them are getting this disease. The father and mother are also not infected. Basically that means it is autosomal recessive trait. Okay, this will be autosomal recessive trait. Whenever you do not see them in the parents, whenever you see like this, and when there's not, the sex is not specified anywhere over here, basically sex is specified, but not everyone has the disease. So just understand you're talking about autosomal recessive trait. And now I have a trick for you to remember. So autosomal dominant one, how do you understand the pedigree chart? So trait appears in every generation. If it is a dominant disease, then it will appear in every generation. This generation also, this generation also, this generation also, if it is a dominant thing. Now, an affected individual has at least one affected parent. At least one of them will be affected for sure. Then only you can get an affected individual. For example, you think both of them are not affected. Okay, both of them do not have dominant trait, obviously dominant allele, then both of them won't be affected. All the progenies will be like this, none of them will be affected. So at least I want one of them to be like this, such that when they bring out new combination, again they could bring the diseased one. So this one will be diseased parent and this one will be the diseased child. So you need at least one diseased parent over there for autosomal dominant trait. Okay. Now in recessive one, traits may skip generation. So you see over here, it is skipping the generation. The next generation you are able to see it, because the recessive alleles A and any small a has to come together. So it might skip the generation. Parents of affected individuals are often carriers. Okay, they might be often carriers, which is why they are unshaded. So they are not showing the disease, they are just carrying the disease. So this is an autosomal recessive trait, my dear children. This came in model question paper 2024-25. Works? I hope you're understanding a bit. Even if you're not understanding, take your time, pause the video and then watch it. But you surely will understand it. Okay? Take it easy. Next, who noted that the behavior of chromosomes was parallel to the behavior of genes. So there was Mendel, okay, there was Mendel if you remember, and Mendel was like, okay, in a pair of factors, he didn't know about genes. So he said, for suppose this is the chromosome and there is capital B, there is small b, then basically he said that in a pair of factors, basically he was saying in a pair of genes, one factor segregates independently of the other. So in a gamete you get this factor, in another gamete you get this factor. Now came two other people who said that basically these factors that he is talking about is not flying around anywhere. It is present on the chromosome. It is present on the chromosome itself, like this. This is the actual thing. So every time a chromosome is separating, obviously these guys are also getting separated. So he said these people, the people who were behind, the question says who are those people who said that the behavior of chromosomes was parallel to the behavior of genes. It was Walter Sutton and Theodore Boveri, my dear children. Direct question. Okay, it came in March 2023. Walter Sutton and Theodore Boveri said this. Works? So they gave the chromosomal theory of inheritance also. Next, female heterogamety is seen in. What do you mean by heterogamety, my dear children? So basically, suppose you have XX and you have XY. This is a female. This is a male. Cool? Now this female, when they give gametes, okay, when they form gametes, the gametes will either have this or this. Either way, the gametes, all of them will be having X only. But over here, in the case of male, it can be either X or it can be either Y. So here I say heterogametes are being formed. Different type of gametes are being formed. This is an example of what? Heterogamety. But over here, who is heterogamous? Males basically. So I see a male heterogamety over here. Whereas the question is asking about female heterogamety. Where do you see it? So this thing, where do you see it? You see it in human beings and you see in Drosophila also. Okay, you see in humans and you see in Drosophila. Okay, there's only one L. And the female heterogamety you will be seeing in birds, where children, this stands for male. So you see male will be always producing Z in the gametes, all the gametes. So male homogamety is seen over here and female heterogamety is seen over here, because every time a female will be producing gametes, it can be either Z or it can be W. Got it? So this is female heterogamety that you see over here. And this is done in the case, this is seen in the case of birds. Cool? So human, Drosophila, they come under this category. Honeybee has a whole different category, we'll talk about that. And then birds is this, when you talk about other insects, okay? For example, yes, in the case of, yeah, insects, should I talk that about here? Okay, fine. You have XX in the case of females, in the case of insect I'm telling, okay, and X naught in the case of males. That means there is only one chromosome, that is X. There is no other chromosome over here. There is no other chromosome here. So basically whenever the gametes are formed, either the gamete will be having an X or the gamete will not be having anything over there. Okay, so though two different type of gametes are also formed over here, which is why both of them, they are example of what? Male heterogamety. Okay, there's only one example of female heterogamety and that is this. Remember that. Next question. The disorder caused due to the absence of one of the X chromosome is? The disorder which is caused when there is an absence of one of the X chromosome. So usually we either have XX or we have XY. Now there is a condition, okay, there is a condition which happens where there will be one X chromosome only. So suppose XX and XY people were meeting and then these are the gametes that are being formed. Okay, so what happened was one gamete was received, the other gamete basically was not received, or something happened, so such that there is no another pair over here, present over here. Get it? There is no other pair that is present over here, which makes this particular particular individual suffer from a syndrome which is known as Turner's syndrome. Okay, what is it? Turner's syndrome, which basically is said to happen to a female. This particular individual, which will be forming with just one X chromosome, will be a female but will have all these sterile conditions. Okay, all the sterile conditions. She would not be a fertile female, she would be sterile with certain masculine features. With certain masculine features, my dear children. Okay, so when there is just one X chromosome, you call this condition as monosomy. There are not any other pair, there is only one pair. Sorry, one chromosome, not another pair, as in there is only one chromosome, there is no pair for it. Fine. Monosomy, and what type of monosomy, children? When you are talking about sex chromosomes, you are talking about allosomal monosomy. Not say autosomal, you call it allosomal monosomy. Okay, so this is in the case of Turner syndrome, and this came in 2023 March. Works? Okay. Next, a patient goes to the doctor worried that a simple cut is taking unusually long time to clot. The doctor identified the patient's condition as what? So, there is a condition where suppose I get a cut and it keeps on bleeding, no one can stop it. So, what is this condition, my dear children? This condition was suffered by Queen Elizabeth also. So, it is known as hemophilia, not was suffered. She is suffering. Okay, so basically it's hemophilia where the clotting factors, the one that is responsible for clotting of the blood, it itself is not being formed properly. So the clotting factors are being affected, which is why the person will not stop bleeding. Okay, so this is the condition, hemophilia, remember that, and it is sex-linked recessive disorder. Okay, I will talk about that later on. I have questions for that also, but if you want, you can remember it. It is sex-linked recessive disorder. Okay, basically not sex-linked, it is X-linked. I can write X-linked. Ayyo, it's not working. Cool. Next question. Which of the following condition of a zygotic cell would lead to the birth of a normal human female? So, in a zygote, what do you need for it to become a female, my dear children? I suppose 2 X chromosomes. Yes. So, we need 2 X chromosomes, then only it will be turning out into a female child, my dear children. Works? Next question. Which of the following is not possible in marriage between carrier woman and normal man? You're talking about color blind people. Okay. So, color blindness, again, it is a X-linked recessive disorder. Okay, just like hemophilia. So, what is basically happening over here? Suppose this is XX female, XY male. Okay, and I'll tell you the general thing that could happen. So, if the person has both the X chromosomes having a mutation over here, okay, both the X chromosome have some mutation over here, which is why this particular person will be known as colorblind. Next condition, she is a colorblind, she is a colorblind female, okay, because XX chromosome is there. Now, there is a condition where this could be XH. That means there is a normal X chromosome and there is another X chromosome which is not normal. Okay, it has some mutation in it. At this point of time, I will say that she is a carrier female. She carries this, but she is not colorblind. Why? Because it has a normal X chromosome which could overshadow its effect and hence make it normal. But she is a carrier female. Now, on the other hand, in the case of male, they just have one X chromosome. So, if this X chromosome is affected, then this male has no choice but to be colorblind itself, because there is no one to overshadow the effect of the X chromosome anyways. There's Y chromosome, but we do not count that. Okay, it won't be overshadowing the effect. So to be a man, to be a normal, you should be like this. So man cannot be a carrier. Okay, man basically would be diseased itself. Man definitely would be diseased. So which of the following is not possible in a marriage between a carrier woman and a normal man? So carrier woman, that means it is XXH, and a normal man as in it will be XY. No, nothing will be there. So what all things are possible, let's see. So the gametes will be formed X, it can be XH also. Here it can be X, it can be Y. Now when you fuse this and this, you get XX. When you fuse this with this, you get XY. When you fuse this with this, you get XXH. When you fuse this with this, you get XHY. Right? This is a female, this is a female. Okay? Here you see it's a normal female. Both are XX. Okay? Here you see she's a carrier female. She is a carrier female. Now over here, XY is there. So it has to be a male, but it's normal male. So normal male, and there is XHY. So here he is diseased. This guy is diseased. So what all can you see? Color blind boys, carrier girls, color blind girls, normal boys and girls. Which one is not possible? It is asked which one is not possible in the marriage. So I can find carrier females. Okay, I can find color blind, whereas color blind boys also. I can't find color blind girls. Do you find color blind girls over here? No one is color blind. One is female is normal, and other one is carrier female. Then I can find normal boys and girls. So normal female is there, normal male is also there. So what do I not find, children? Color blind girls. Only carrier girls are there, color blind girls are not there. Got it? This is how you need to solve this. Cool? So please understand this funda, then only you will be able to do the questions. Works? Next question. Tall structure, undeveloped testes and gynecomastia are what? Are found in which disease or which syndrome? Basically tall structure, underdeveloped testes, that means this is, you're talking about a male only, but the male is having these problems. And gynecomastia, what do you mean by gynecomastia? Gynecomastia means what? Development of breast. So if it was a female, then development of breast is also quite normal, but this is a male, right? So in male, when something comes, you're talking about Klinefelter syndrome. You are talking about Klinefelter syndrome, my dear children. Okay, so Turner syndrome is when you're talking about females. Turner syndrome is when a female, instead of having XX chromosome, is just having X chromosome. This XO condition that is being found. So I say X, there is only one X. So how many number of chromosomes do they have in total? Usually we have 46 number of chromosomes. 23 pair means 46 number of chromosome. But over here, she will be having what, children? She will be having, oh, did I actually remove it? No. She will be having just, 45 chromosomes. There will be only 45 chromosomes. If I want to divide properly, then it is 44 autosomes and 1 X chromosome. 44 autosomes plus 1 X chromosome. That is in the case of Turner syndrome. In Klinefelter syndrome, what is happening? Basically, it should be a man, so it should be XY. This is a normal pair, but there is an extra X which is coming around. So probably, if this is a female, this is a male, here 1X should be coming and here XY. Here also, there should be either Y or it should be either X. Suppose both of them are coming and joining. So you get XXY. Or from here, you get X, 2X and 1Y over here. Anyways, at the end, you should be having XXY. There should be one X coming around over here, which is Y. This particular male has female characters as well. That is what? Gynecomastia, an undeveloped testis and what? Tall structure. Okay, so how will you write how many chromosomes are there in total here over here? 46 is normal, one extra is there. So what do I say? 47 chromosomes are there. Okay, if I want to divide, then I'll write 44 autosomes are there. okay, the normal chromosomes which is not sex chromosome, and then I have XXY, and then I have XXY. So 44, 45, 46, 47 makes total of 47. You get it? Okay. And this is known as, this is also allosomal disease, since it is sex chromosome that is getting affected, and you have one extra, so you call it allosomal trisomy. So instead of having a pair, you have one extra, you call it trisomy. When you have one less, you call it monosomy. Allosomal monosomy, allosomal trisomy. So you're talking about Klinefelter syndrome over here. Works? Done? Done with the 10 MCQs that we are having. Let's move on to the next questions. So write a short note on sex determination method in birds. In birds, how can you write a short note on in the case of birds? So I told you in the case of birds, okay, they have gametes like this, okay. So they have ZZ on one particular organism and ZW in another particular organism. This is going to be a male and this is going to be a female. Do not worry, I have the answers already written, you just have to read it. I will be showing you that as well. I'm just explaining things to you, okay. So basically over here, this male can contribute to type, basically this male can contribute to gametes, both of them will be the same. It is homozygous condition, homogametic condition. Over here it can be Z and it can be W. Okay, so this is what the parent. You have to make like this. This is what the gamete, and this one will be F1 generation. With this and this fuses, you get Z. Okay, when this and this fuses, you get ZW. When this and this fuses, you get Z. When this and this fuses, you get ZW. So let's see who all are male and who all are females. ZZ, so it has to be a male. ZW, so it has to be a female. Again ZZ, it has to be a male. ZW, it has to be a female. So can I say 50% of them are male and 50% of them are female. Works? And how will you write this as well? So let's see. So two different type of gametes in the terms of sex chromosomes are produced by the female, that is female heterogamety. You have to write like that. That basically in the case of females, they are producing what? Two different type of gametes, and this condition is known as female heterogamety. Yes, two different type of gamete you are getting. No, here you are getting same type of gametes. Now in this type, female is heterogametic whereas the male is homogametic. Written. Next point, the female produces two type of X carrying Z and W chromosome, whereas the male produces same type of chromosome. If the sperm carrying Z chromosome fertilizes with the egg carrying Z chromosome, the zygote develops into a male, right? And now the sperm carrying Z chromosome goes and fuses with a egg carrying what? W chromosome, will be developing into a female. This is how you can write and this is how you can draw also. But here they have mentioned autosome, so I don't want you to do that. You can just make this. But please make sure you are mentioning about parents, gametes and F1. So whenever you are writing answers, underline the important terms, that is female heterogamety, like this. Okay, it's very, very, very important. Will you be able to answer like that? Okay, so I thought I will put down the answers for you to refer. It becomes easy, right? Next thing. What is haplodiploid sex determination mechanism? Mention an animal which exhibits this. I'll say honeybee. I'll say honeybee exhibits this. And what happens over here? What do you mean by haplodiploid sex determination? Do you understand haploid and diploid? So diploid is when you have 2n amount of chromosome. That is, for example, if n is suppose 16, okay, so 2n will be 32, double the amount of chromosome and everything will be there. Okay, this is what? This is diploid, 2n, 32, whatever you want to say. Okay, now haploid if you want to say, that means it's just n, half of it, it will be just n. So n is what, children? N is equals to 16. Remember that. Now we are going to do the thing. So females, I am saying they will be having 32 number of chromosomes. Okay, over here I am saying males are the ones that will be having 16 number of chromosome. Now when the females, they undergo meiosis to form gametes. Okay, and meiosis, what happens? In meiosis, what happens? For example, this was a cell that was having 32 number of chromosome. Now this cell will be undergoing division, that is meiosis, where at the end the number of chromosome becomes half. So here she will be giving one egg with 16 chromosome, another egg with 16 chromosome. Okay, male since they are already 16 in number, they cannot make it half. They do mitosis. So here I see meiosis for gamete formation, but here I see mitosis. They are just increasing the number. So suppose 16 itself is there over here. Now, this particular male gamete, if it fuses with the female gamete, 16 comes together to give me 32. And 32 is found in males or females, my dear children? It is found in females. So this particular zygote will be turning out to be a female. Then what about this, ma'am? This also can fuse with another 16. No, that does not happen. That is the thing. So basically this thing will undergo some other process which is known as parthenogenesis. That means without fertilization only you are going to make a zygote. Without fertilization, without fusion, you are going to make a zygote. So that is a scientific process that happens, parthenogenesis, where now this particular thing will be turning into a male. That's all. It will be having 16 only, without any fertilization happening. There is no fertilization, please remember. Without fertilization, you are forming a zygote, and this zygote is going to turn to be a male. Now you tell me, is this haploid or diploid? This is 2n, so this is diploid. It became half for formation of gametes. Now you have haploid gamete over here. Similarly, this was already haploid, so it didn't undergo meiosis, because if it underwent meiosis, then it will be half of it. So it didn't undergo meiosis, it underwent mitosis, and now you have 16 only. So this is still haploid. So a haploid comes together to give 32. This is again what? Diploid. But what about this guy? This is still haploid itself. This is still haploid. So the haploid one will turn to a male, the diploid one will turn to a female. This is what you mean by haploid-diploid sex determination, and this is exhibited by honeybee. I know this is unbelievable thing that is happening, but it happens as well. So there is no choice but to agree to this fact. So the haploid-diploid sex determination, how you have to write the answers, check, okay, is a system in which the sex of the individual is determined by the number of chromosome set it inherits. Okay, so if it is double the number of chromosome sets, basically it's okay. If it is half, then it will be male. So males develop from an unfertilized egg and are haploid, having a single set of chromosome. Okay, females develop from fertilized egg and are diploid, having 2 sets of chromosome. Fine. Now a queen bee, how does it work? So queen bee basically lays egg. If an egg remains unfertilized, it develops into a male drone. So basically the males which are being produced now, what do you call them? You call them drone children. You call them drone. Okay, the female which is produced here can be a worker bee or it can be a queen bee, but at a time there will be only one queen bee. Okay, rest they will be all be turning out to a worker, and there will be a queen bee who will be laying eggs and doing things. Okay, so basically over here you have to understand one very important fact. Worker bee, what do? They work around their comb and honeycomb and all. They make sure everything is fine. They make the royal jelly, things like that. Okay, drone, it has just one function, and that is to mate. So during a flight which is known as nuptial flight, the queen will be moving around up, okay, for fusion. Okay, similarly the drone will be also, there will be fight happening between so many drones and the queens to get the queen. Okay, at the end they will be fusing, only one will be fusing, and that one will be dying also. That drone will be dying also, but it happens, it has to happen. That's how things work. So the males, all of them, they are drones, my dear children. So what is haploid-diploid sex determination mechanism? It is where the sex of an individual is determined by the number of set of chromosome that it inherits. The males will be developing from unfertilized egg. It will be having half the number of chromosome, or basically you can just say a single set of chromosome. That will be much more correct. Okay, it will be having just a single set of chromosome. It is known as diploid. Now females will be developing from the fertilized egg and would be having diploid thing, that is having 2 set of chromosome. If I said diploid first also, please forgive me, it's haploid and this is diploid. Works? So until here you can write the answer. This will be much more than enough, and please make use of this also. Fine. You have to show it like this, much more cleaner picture, but you have to show like this, then only things will be working. Next, explain the sex determination in honeybee. There is one more question which was asked. So this was asked in the model question paper. So this time model question paper again gave you a question like this, but they have reframed the question. Before it was like this, 2022 July. Explain the sex determination in honeybees. That is a direct question. Over here they have spoke about haplodiploid thing. So this is another answer which you can refer for answering. So if you want, you go have a read for it. Okay. The female is diploid having 32 chromosome, the male is haploid having 16 chromosome. An offspring formed from the fertilization of a sperm and an egg developed into either a queen of workers, both are female. An unfertilized egg develops into a male by means of parthenogenesis. That method of sex determination is known as haplodiploid sex determination. Male produce sperms by mitosis and female produce eggs by what? Meiosis. They will have the number of chromosomes, right? Hence the male honeybee do not have father and thus cannot have sons, but they have grandsons. So what do you mean by that? That they do not have grandsons? This is there in your NCERT. I'll explain you. So listen, basically this guy, if you see over here, this is a male. It does not have a father because nothing came and fused over here, but it will be having a grandfather because obviously 16 plus 16 has to come together to give 32. So obviously there was a grandfather, but over here it does not have a father. This is a thing that is given in your NCERT. This is not something that I am making up. Okay. So this is the 13th question, which is the exact repetition of the thing that we have studied. Next thing, write the genotype of the parent when their children are with A, B, O blood groups. What will be the genotype? Genotype means genetic makeup of the parent. So I'll tell you one thing, okay. This is, you have 3 alleles for blood group. One is IA, which is for A blood group, one is IB, which denotes for B blood group, and one is IO, or basically you write like this, which denotes O blood group. And I am telling you, A and B, they are equally dominant. Okay, so if both these genes come together, it will be AB blood group. Okay, now A comes with O, A will be dominant, B comes with O, B will be dominant. So I understand that this guy is recessive, both of them basically are dominant. Okay, now I want these many blood groups in the progeny, in the children. So what is the thing that I should be having? I should be doing, what will be the parental combination? Suppose I have IA and then IO over here. I cross it with another thing, that is IB and IO. Okay, this is one parent, this is another parent. The gametes will be IA, IO, here it will be IB, here it can be IO. Remember, this is parent, this is what? Gamete. Write it properly in exam, okay? And then this is F1 generation. So what do you get? This and this fuses, you get IA, IB. Okay, that means the blood group is what? A and B both are there. So which one is dominant? Both of them are equally dominant, so it will be AB blood group. Okay, now this and this comes together. So I get IA, IO. That means A is dominant over O, so it will be A blood group. Okay, now this and this fuses, I get IO, IO, so that will be O blood group. Then I have this and this, which will be again, this and this, so it will be IB and IO. That means what, children? Wait. Did I do something? Okay, sorry. When this and this comes together, you get, when this and this comes together, you get this. Okay, when this and this comes together, you get this. And just rearrange it on order. Okay, this will be B blood group. Why? Because B is dominant over O. So how many blood groups we got? A, B, A, O, B. What was the question? What type of parents would be having these type of blood groups? So this is the parental combination. Now suppose on the other hand it was AA, or it was IB over here. All the progenies, whatever you do, all of them will be AB only. We won't be getting A, B and O. So if you want O, if you want A and B also, so just understand that this is the thing. This is IA, IO, IB, IO. Then only you can get it. Works? So the answer for this is, the genotype of the parent blood group is IA. This is basically IO, the small I. Okay? And IB, IO, either way. And you have to show it also. This is not enough, you have to show it like this. Make this and then show it. Okay, and you just have to rearrange this If you're finding it difficult, if you think that ma'am has made us confused, so this and this gave you this. Oh, sorry. Again, ma'am is doing something really bad. Okay, this and this gave you this. This and this gave you this. This and this gave you this. And this and this gave you this. Works, but you should be writing this first and this first. Okay? Works, but write it like that. Next question. Write 2 symptoms of Down syndrome and write the chromosome number in Down syndrome. What do you mean by Down syndrome? This is a very important question, children. All the chromosomal diseases that I taught you right now, it's very much important. Klinefelter's, Turner's. Now Down syndrome. So Down syndrome, it has nothing to do with the sex chromosome. Okay? It is autosomal trisomy. It is what? Autosomal trisomy. That means what? This time you are going to add up chromosomes in the autosomes. So there is a 21st chromosome pair, that is this. This is a pair of 21st chromosome. And what is exactly going to happen is that there will be one more chromosome being added up here, which is why it will lead to trisomy. There's a pair already, you have one more extra thing which results in trisomy. So suppose we always have 46 number of chromosome, out of them 44 is autosomes, and then X and Y, or basically X and X or whatever, 2 other sex chromosome will be there, making it 46, 44, 45, 46, right? This time you have one extra autosome, so it will be 45 plus whatever, it can be a girl child or it can be a boy child, which will be having Down syndrome, my dear children. So please understand one very important thing. Down syndrome, Klinefelter syndrome, and what Turner syndrome, everywhere you see addition and deletion of chromosome. So all of them, they are known as chromosomal disorders. All of them, they are known as chromosomal disorders, or you call them non-Mendelian disorders, or you call them non-Mendelian disorders. Please remember it. Mendelian disorders are the one which arises when, for example, this is a chromosome, on this chromosome there is a gene which is getting mutated. So whatever condition would be arising because of this will be a disease, will be causing a disease, and this disease is known as Mendelian disorder. Okay, for example, hemophilia, a gene responsible for clotting factor was getting mutated, which is why you had hemophilia, or any other thing, sickle cell anemia, whatever it is caused like that only. So you have to understand the chromosome number will be for Down syndrome will be 21 and will be trisomy of autosomal chromosome, my dear children. Now what are the 2 symptoms of Down syndrome? They will be having a broad palm with significant palm crease. Palm crease as in you can see the lines, no, they will be having very darker, very different type of lines than us. Okay, mental retardation is something that can be seen in them. The people who have Down syndrome, they will be having mental retardation, and there are other points also. This is a very important question, you have to remember it. The tongue, if you see, they have furrowed tongue. The tongue is kind of furrowed tongue. That is also slight thing is there, but yeah, it is there. And partially open mouth and all this things. So let's see. Individual is short statured with small round head. So people with Down syndrome, if you have seen them, they have a small round head, and their height is also not very much. Okay, so individual is short statured. Try to remember things rather than mugging things up. So furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical psychomotors and mental development is retarded. Psychomotor, basically, for example, I need thinking and working skill also. For example, I have to think and I have to sue. Both of these things won't be working together for me if I am a person who has Down syndrome. So physical psychomotors and mental development is retarded. Children, the chromosome, if you're talking about, chromosome in number is 21 chromosome. The karyotype, as in the chromosomal makeup, if you're talking about, the karyotype of Down syndrome individual is 45 autosomes plus XY. If it's a male, or it can be a female also, either way. So in total, how many chromosome? 47. Similarly here also 47 in the case of females. Either way it can happen. So you have to write exactly like this only. Okay, it can be 45 autosomes plus XY, or it can be 45 autosomes plus XX if it's a female. So out of this you can write 2 symptoms and then you can explain this part of the question. Works? Now this question is very much important. I am telling you again and again, please trust me on that. Now, next question. Define point mutation. Give an example for point mutation. Point mutation is basically when there is only a single nucleotide which is getting mutated. For example, I hope you know that DNA basically is responsible for making proteins. But the one who make proteins is outside the nucleus, and DNA is where, children? Inside the nucleus. So, the one who make protein is ribosomes. So, the information to make the proteins is given to something else that is known as mRNA. So, mRNA is being found from the DNA, and this mRNA moves out and comes between the ribosomes to form the proteins. Now, how it is read, children? Usually in the form of codons. So, suppose this is a chain of mRNA and there are codons, multiple codons. I am writing GAG and so many other things. My thing is GAG. So, over here, suppose due to some mutation, instead of GAG, which was coding for an amino acid that is known as glutamic acid, now there has been a replacement. There is a point mutation happening at one particular area, so that A is replaced by U, my dear children. A is replaced by U, and now since it has become like this, it will be GUG, and it will be coding for something else such as valine. Okay, now it will be what, children? Valine. So, over here you have GUG and it will be coding for valine. That is a problem, because you know what, amino acids, they have to come together to make a protein. Now, one amino acid, if it's changed from this to this, obviously the protein which is being made will also have some problem. Now, this is the condition of sickle cell anemia. Sickle, my God, what happened to my spellings? Okay. Sickle cell anemia. Children, I hope you are understanding. I am trying my level best to make you understand things. Okay. Definitely boost up my confidence also if you are understanding. Tell, yes ma'am, I am understanding, and if you have any doubts, definitely ask that also. So, sickle cell anemia is where this is being mutated such that now you have GUG, and this particular protein which was, this particular thing which was going to make a protein, what protein was it, children? Beta globin chain. It was beta globin chain, and if you are aware, in the case of hemoglobin, we are actually wanting to make something that is known as hemoglobin, and in hemoglobin you have two globin chains, one is alpha and the other one is beta. Now, if you are altering beta, then obviously do you think this hemoglobin will be a normal hemoglobin? No. It is not a normal hemoglobin that is going to be formed, which is why the cells which carries this hemoglobin will ultimately change its shape to sickle cell, because it is not carrying a normal hemoglobin, and now it will be sickle cell, and this sickle cell obviously would be transporting very less amount of oxygen and things like that. Okay, so there are sickle cell anemia carriers, and there are people who got two of the genes from both the parents, such that they will be a diseased person and they would be dying as well. So there are carriers who would be carrying this thing, who would not be showing any character in them because they have a normal allele to cover things up. Okay, so basically HB, okay, it's a normal allele. Suppose this is affected, another allele is also affected, both of them are affected, then there is no one to overshadow the effect. This particular person will be having sickle cell anemia. But if one is normal and if one is normal and the other one is not normal, then there is one to overshadow it. So hence it will be a carrier. Okay, so this is again an autosomal recessive disease. It is an autosome, it's not sex linked, like hemophilia was sex linked, right? Remember, color blindness is also sex linked. It is always on the X chromosome. Over here it is not like that. It is autosomal recessive disease. So in recessive condition, when both of them come together, that's only when you will have that disease, otherwise you will be a carrier. So this is an example of point mutation. Just because of small mutation that is happening over here, you get a whole disease. So how can you write the answer? Change in a single base pair. So I hope you know that these things are known as base pairs. A base pairs with T, or basically U, G base pairs with C. So these are base pairs. So if there is a change in one such base pair, obviously it will lead to the whole trouble. So change in single base pair, please remember, single base pair of DNA is known as point mutation. A classical example of such mutation is sickle cell anemia, where in the codon GAG, the second nucleotide, that is A, is replaced by U, hence making the codon as GUG. And how do you remember A is replaced by U? A comes before U, You can remember that A comes before U, so basically A comes before U, so A was there usually, but then later on U came and replaced it. Now glutamic acid G comes before V, so glutamic acid was there before, but valine is coming and substituting it. This is how you can remember it. Okay, works. So note that GAG codes for glutamic acid, whereas GUG codes for valine. Next question. What are the conclusions drawn by T.H. Morgan from the crossing experiments in Drosophila with respect to linkage? So when he did the experiment, okay, he wanted to find out how does recombination and everything take place. So what was his conclusion at the end? T.H. Morgan and his student was also there. His name was Alfred Sturtevant. Okay, Sturtevant. So there were two guys who were working on it. Okay, and I told you in the initial part of this particular session that basically if the genes they are closely linked. If they are closely linked, will they show much recombination, my dear children? No much recombination. But if the same genes they are in distance, they are loosely linked. If they are loosely linked, then they will show very high amount of recombination. Works? And we can see that in the form of writing. The higher the linkage, lesser is the recombination. Linkage as in the closeness, okay? Higher the linkage, lesser is the recombination, and the lesser the linkage, higher is the recombination. In other words, genes which are tightly linked will show lesser recombination, and the genes which are loosely linked will show higher recombination. You can take the example with the help of pictures also to make them understand things, okay? It's 2-marker question, so you can explain like that. Works, and please write in points as I have shown you in each of the answers that I have shown you until this particular time. I have written in points for you to understand that writing in points is very much important, my dear children. And underline wherever you see, for example, lesser the linkage, higher recombination. Higher linkage, lesser recombination. Okay, so when it's a 2-marker question, you don't need to underline very much so that you underline the whole thing. But please underline the linkage, recombination. At least you can do that. Works? Okay, next, what is a test cross and what is its significance? So for example, I say that I have a plant, tall plant. I have a tall plant, children. Okay, but the problem is, tall can be something like this and tall can be something like this also. Because here both the gametes are there, both the alleles are there and both has to be tall. Okay, they are dominant, right? This is the same type of alleles. Over here, this allele is dominant and this is recessive, so this will overshadow the effect and this will also become tall. But with seeing, I cannot understand whether it is this or this. So whether it is homozygous tall, I cannot understand whether it is homozygous tall. Homozygous as in same. Okay, same alleles are there. Or it is heterozygous tall. So to understand this, what can I do is, I can take both the plants, I can cross it with a recessive parent. I can cross with a recessive parent. Ma'am, what's the use of that? Let's see. So I'm going to talk about that. Let's cross this guy first. So you have capital T, and recessive parent means both have to be like this. The gametes, this is the parent, this is the gamete. Please write parent and gamete in the exam, okay? So this is capital T in a gamete, this is small t and small t in another gamete, okay? Now F1 generation, let's see. This and this fuses, you get what? Capital T, small t. This will be tall. This and this fuses, you get capital T, small t. This will be again tall. This and this fuses, capital T, small t, again tall. This and this fuses, capital T, small t, again tall. All of them, if you see, they are tall, my dear children. Only if what? Only if the condition is like this. If it was homozygous, then only it will be tall, because you are going to see the difference now. Suppose this is capital T, small t, and then you are crossing with the recessive parent. Over here, this is the parent. The gametes that will be formed will be capital T, small t, small t, small t. F1 generation, when I fuse this with this, I get capital T, small t, agreed. When I fuse with this, capital T, small t. So both of them I see they are tall. Now let's see the other two. This and this, you get small t, small t. This and this, you get small t, small t. Both of them, if you see, they are dwarf. So 50% of them are tall, 50% of them are dwarf. So if I do this experiment, if I get the progenies, 50% of them are tall and 50% of them are dwarf, I understand that basically this tall plant is actually heterozygous tall. This is what you mean by test cross. I am doing a test over here by crossing with what? A recessive parent, my dear children, to find out whether it is homozygous tall or heterozygous tall. I want, if for example, I want pure breed plants, that means it should be dominant, both of the alleles should be same like this, and if I want to find that, then I have to do this experiment. Why do you think I need it? Because I can use them to create more tall plants. If I am using them, I get only 50% of them to be tall, rest of them will be dwarf. So I want this to happen, which is why I did this. So it is a kind of back cross where F1 plant is crossed with what? Recessive parent. It helps in finding F1 plant is homozygous or heterozygous in genotype. Works. So the same example is given over here in the form of Punnett square. So instead of writing like this, they have made it in Punnett square. How does the Punnett square work? In the same thing, for example, so what do you do is, you write the gametes on one side, for example, capital T, small t. Here the gametes on another side, small t, and then fuse. So it will be capital T, small t, here it will be capital T, small t, here it will be small t, here it will be small t, small t. Works. So if we see, two of them, they are tall, two of them, they are dwarf. Works. Is the 18th question. Next. What is phenylketonuria? Write the cause and symptoms of phenylketonuria. Now, if you are a person who is finding it difficult to remember this, then you are at the right place, my dear children. I am going to make you memorize this very easily. So, in our body, there is an amino acid which is known as phenylalanine. What do you call it? Phenylalanine. I hope, children, you are understanding the way of teaching. I am trying to teach you the concept first so that it does not look like that I am directly teaching the answer. I don't want you to do that. I want you to understand the concept. It becomes easy for you to frame your own answers as well. Yeah. So I hope it is helpful for you. So over here you have an amino acid which is known as phenylalanine. Okay. And what happens is that there is some enzyme which will be converting this phenylalanine to very important thing which is known as tyrosine. Okay, and who is that enzyme, children? Its name is phenylalanine hydroxylase. Phenylalanine hydroxylase. Now you know this is what, children? This is an enzyme. And enzymes are nothing but what, children? Protein. And proteins have to be coded by what? Genes. But what if the gene which is coding for this is mutated? What if it is mutated? Then the process, the whole process of making this into a protein, making it into an enzyme and then making it into a final form will not be happening, and hence the whole process will also not be happening. So you are not converting phenylalanine to tyrosine. Instead, what is that something that is going to happen? The Dracula is going to come over here. Basically, phenylalanine, okay, is going to convert into phenyl pyruvic acid. It is going to convert into phenyl pyruvic acid, which is the Dracula. Because if it stays in our body, it gets deposited in our brain and our blood, and cause what? Mental retardation, my dear children. It causes what? Mental retardation because of the accumulation of this particular thing. This is what you mean by phenylketonuria. The disease that you get at the end is known as phenylketonuria. So you know the cause of this, and what is the symptom? mental retardation and things like that. Okay, can we see the answer? Let's see. So answer is, it is an autosomal disorder due to the presence of a recessive gene. First thing, it is an autosomal disorder, it's not X-linked, nothing like that. It happens normally in an autosome, a normal chromosome. Some gene is there which does this, but it is mutated. Okay, it is an inborn error of metabolism. Okay, it is inborn, children. It is genetic, inborn error of metabolism and inherited through autosomal recessive trait. It is autosomal recessive trait. So as of now, you have different autosomal diseases, recessive diseases. What are those? You have sickle cell anemia. Okay, apart from that, you have this thing, phenylketonuria, and you have something that is known as thalassemia. That is also same autosomal recessive diseases that you are having. Okay, X-linked recessive disease, hemophilia and color blindness. Okay, so the affected individual lacks an enzyme phenylalanine hydroxylase that converts the essential amino acid phenylalanine, a substance found in a food, to what? Tyrosine. As a result, this phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives, which is not really good for us. Symptoms: accumulation of this in brain results in mental retardation. Phenylpyruvic acid and other derivatives also excreted through urine, which is why it is known as phenylketonuria. Okay, because of its poor absorption by the kidney. Okay, phenylpyruvic acid is not taken up by the kidney, which is why it is removed in the form in the urea, it's in the urine itself. Okay, so yeah, infants have lighter skin, hair and eyes than the siblings. Okay, why do you think the pigmentation of the color is also, pigmentation of the skin and the eye is also affected? Because I'll tell you something very important which is given in your book. Okay, so usually what happens is that one gene is there, one gene always is responsible for one trait. That is, for example, you have capital T is for height. But sometimes what happens, the genes which is there over here, it will be responsible for multiple trait. So trait 1, trait 2, trait 3, things like that. So this particular gene, which is coding for this particular enzyme, what it is actually doing is that it is forming tyrosine initially, right? And this tyrosine is helping in the formation of something else which is known as melanin. And melanin is responsible for our skin and hair pigmentation. So if tyrosine is not there, melanin is not there. If melanin is not there, our pigmentation also goes low. So which is why the people who have this disease, indirectly they will be having this condition also, that is known as hypopigmentation. Hypopigmentation, not gigmentation. Wait. Hypopigmentation. Okay, so mental retardation you have to remember, and in the urea it's also removed, and then they have lighter skin also, my dear children. Works? It is autosomal recessive disorder. It is inborn error. Okay, it is not something that you gain throughout your lifetime. It is genetic, and all the genetic diseases come from the childhood only. So all the Mendelian disorders are going to come from the childhood, when you moment you are born. Done. Let's go with the next question. So T.H. Morgan selected fruit fly for his genetical experiment. Why? Give any 3 reasons. Why did he choose fruit fly? Because basically what experiment was he doing, children? The linkage and recombination one, along with his student, that is Alfred Sturtevant, right? So why did he actually choose fruit fly for that? That is Drosophila melanogaster for that. Why do you think? Basically because of these reasons. They could be grown on simple synthetic medium in laboratory. Okay, it was very easy to grow them. Okay, they complete their life cycle in about 2 weeks, so they you don't have to wait for an year to see the result. You can see them in the life cycle, that is about just 2 weeks. A single mating would result in what? Multiple offsprings. Okay, and they had a clear differentiation of the sexes, that is the male and the female flies. So it was very easy for them to mate, make them mate, and then produce things. So what are the things, children? It was easy to grow. Okay, it had a very short lifespan, that is about 2 weeks. Then the number of progenies that was being produced were also too much, and the male and the female sexes were distinguishable, so you could easily distinguish it. Now what are chromosomal disorders? Chromosomal disorders arise when you have a gain or a loss of chromosome. Okay, when you have a gain or a loss of, it looks really bad, just a second, I'm so sorry my dear children. gain or loss of chromosome. Actually, my hands are paining. Okay, which totally disturbs the chromosome number in an individual. So what are the diseases? I told you Down syndrome, trisomy. What trisomy? Autosomal trisomy. Okay, then you have Down syndrome, then Klinefelter syndrome, that is what? Allosomal trisomy. Then you have this thing, what do you call it? Turner syndrome. What is it? Allosomal monosomy. So over here you see gain and loss of not electrons, oh my god, chromosomes, which is why you get this disease. Okay, I have slowly started liking chemistry, I think. All of a sudden I am missing Aishwarya ma'am. Yes, so disorders in human beings are caused due to absence or excess or abnormal arrangement of one or more chromosomes are called chromosomal disorders. So it can be gain, loss or abnormal arrangement of chromosome, which are known as chromosomal disorders. Example: Down syndrome, Turner syndrome, Klinefelter syndrome, which is there in your NCRT. Works? Cool. So with this, I think we are completing the 20. So basically 20 questions and 2 marker questions. All the 2 marker questions are finally completed, my dear children. So we should be having a big tap on our back. Do that, do that. You deserve it. Okay. Now the 21st thing that I am having over here, at the end of the session, I will be talking about the most important topics from this particular chapter. By far, I think you have already got it. You saw how many number of times different questions have been framed around the chromosomal disorders or the Mendelian disorders, linkage, recombination and things like that. The sex determination in different type of organism. You had honeybees, you had human beings, you had fruit fly, this fly, that fly, insects and everything. So you know the important topics as of now. I have chosen those topics and you see the repetition as well, what all type of questions are coming. So you should be understanding that. But I will be anyways telling you at the end, so do not worry about that. Let's solve the 5 marker question. Let's begin. There are 5 marker. We will solve it soon. So, few autosome linked recessive gene blood diseases occur in human population. Blood diseases. Okay. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Explain. So it is autosome linked gene recessive disorders. Okay, that means it does not have to do sex with anything with the sex chromosomes. Fine, it can happen with anybody, you, me, anyone. Fine. So there are 2 such diseases. One is sickle cell anemia. I'll explain first and then I'll show you the answer. Okay, and the other one is thalassemia. What happens is that both of them are going to have a problem with hemoglobin protein. Hemoglobin protein. So the thing is, in sickle cell anemia, the gene which codes for beta globin is affected. Okay, and over here, as I said, it was initially GAG, but slowly now it has been replaced by GUG. There is a replacement over here. Okay, and hence now instead of having glutamic acid, it will be what? Valine. And this is happening in the 6th codon. This codon that you see is the 6th codon. 6th codon, as in similarly you can find 5 more other codons over here. Okay, this is happening in the 6th codon. 6th codon, 2nd position change is happening, which is why you get this. Now over here, this particular gene was basically coding for what? Beta globin chain. Now still it is coding for beta globin chain. But the problem is that this beta globin chain that you see over here, it is defective. Okay, it is defective. Still you will be forming hemoglobin. Okay, but the quality of this hemoglobin is going to get affected. Quality is going to get affected because the quality of this is compromised, children. Please remember, quality is getting affected, not quantity. Still beta globin chain is being produced as it was produced, but the quality is getting affected. Okay, so I will say that sickle cell anemia is a qualitative disorder. Qualitative disorder. Works? Now, when you talk about thalassemia, things are quite different there. So, in thalassemia, one thing before that I should be telling you is that hemoglobin is made up of alpha chain and beta globin chains. So, beta globin chain was the one that is getting affected in sickle cell anemia. Okay, but in thalassemia, things are quite different. Alpha chain can be affected, beta globin chain can be affected. For formation of alpha chain, you have HbA1 protein and HbA2 gene also. Here you just have HbB. There's only one. Okay, there is only one gene with 2 alleles. This will be having 2 alleles, children. This will also be having 2 alleles. This will also be having 2 alleles. So basically, one allele of this gene, if it is getting defected, then basically you are getting lesser amount of alpha globin chain. Similarly, one allele of this is getting affected, you are getting lesser. Suppose both these alleles are affected. Still there is one more gene to code for it. This will definitely produce it, but the amount of alpha globin chain that will be produced, the quantity if you see, it will be really less, because this is getting affected, it is not producing thing. Still this is producing, but not enough. Similarly, if one allele is working, another allele is not working. This is not working, this is working. So still this is producing beta globin chain, but the amount is kind of really less again. The quantity is what? Is less. So that is a problem. Okay, so we say that a person is having thalassemia. It can be a minor, it can be a major patient also. Minor patient as in, yes, there might be a problem in one gene, but still production is happening. Here also some gene might be affected, some allele might be affected, but yes, production is happening. Okay, so we say it is minor case. But when suppose altogether this gene is, both of these genes are not working, that means you are not making alpha chain. That is a big problem. So this is a major case. Similarly over here, beta gene is not being made. That is a big problem. So that is a major case. So we say major thalassemia and minor thalassemia patient. Get it? So this is a quantitative problem, whereas when you talk about this is a qualitative issue. You're messing up with the quality, here you're messing up with the quantity itself. So both of them, they are autosome linked recessive disease. You will get this disease when both the genes will be having the mutation. When both the genes will be having mutation, then only you will be having this particular disease. For example, it should be HB, HBS. Both of them should be having this mutation. Then only you will get this disease. Similarly, both of the alleles should be having this, then only you will get thalassemia also. Now, how you have to write this answer? Like this. So, some blood diseases in humans are caused by mutation in autosomal genes, leading to issues with synthesis of blood proteins. These problems can be categorized as qualitative, affecting the structure of the protein, or quantitative, affecting the amount of protein being produced. So here you are not producing enough amount of alpha chain or beta globin chain. Okay. Now sickle cell anemia is one example, that is a qualitative problem. Qualitative. Okay, you are messing up with the quality. Mutation in HBB gene on chromosome number 11 causes production of abnormal hemoglobin, that is HBS. Okay, that is HBS. Now over here, this defective hemoglobin leads to sickle shaped red blood cell, impairing oxygen transport and causing blockages in blood vessel. Hence the defect is caused because of the error in the quality of the protein that is being made. Okay. So abnormal hemoglobin is HBS, which you write like this, HBS. Fine. Now next, thalassemia, quantitative problem, caused by mutation in gene responsible for hemoglobin synthesis. Okay, you have HbA1, HbA2 for alpha chains, or HbB for beta chains. Over here, there can be, just a second, yes. There can be mutation in one allele of a gene or both alleles of a gene. So as I said, it can be mutation in one allele. This gene can have multiple alleles also. Similarly, this gene will have multiple alleles, that is 2 alleles. Here also you will have 2 alleles. So it can be mutation in one allele or both the alleles. Okay, which leads to reduced or no production of specific hemoglobin chains, causing anemia and fatigue. Hence, the direct defect arises due to errors in the quantity of the gene produced, or basically I can write quantity of the proteins which are being produced, my dear children. Just give me a second. I'm not able to see because I touched somewhere. Yeah, now it works. Okay, I hope you got it. So you have to write the initial part. The problems can be categorized as qualitative, that is affecting the structure, quantitative, affecting the proteins that are being produced, amount of protein that is being produced. Works? So this is a 5-marker question, answer it properly. Next, give reasons for the following. What are the reasons for the following? Let's see. So this is a question from model question 2024-25. A simple cut results in non-stop bleeding in hemophilia affected individuals. Why, children? Because the clotting factor gene is getting affected. Clotting factor gene is getting affected. If clotting factor gene is getting affected, do you think that it will be producing thing? No. So you have to write that hemophilia is a sex-linked recessive disorder where the clotting factor gene is getting mutated, which is why there is no clotting and hence there will be non-stop bleeding. Turner syndrome affected females are usually sterile. While they are sterile, children, because they just have one chromosome, one X chromosome, which is the reason why they have sterility. Okay, they are sterile in nature. They do not have normal XX pair of chromosomes like a normal female. Okay, now in Morgan's dihybrid cross experiment on Drosophila showed that flies having genes for yellow body and white eyed exhibited less recombination. So if less recombination is there, that means what? The genes which were there, the genes for yellow body and white eyes, it will be closely linked or loosely linked. If less recombination is there, it has to be closely linked, or what do you say? Tightly linked. Which is why you see less amount of recombination. Now, inheritance of skin color in humans show different phenotype. Inheritance of skin color in humans show different phenotypes. Why? That's an interesting question. So every time I told you there is a gene, there is a trait, right? But now I'm going to tell you something else. There is a trait and I'm going to tell you that this gene, all of them comes together, works around it and brings out traits. So suppose I have capital A here, capital B over here, capital C over here. When these genes come together, I say that this individual is having this genotype. So I'll say this individual will be having darker skin tone because all of them are dominant alleles. On the other hand, if I say it is capital A small a, one gene, capital B small b and capital C small c. Together it will be bringing capital A, capital B small b, capital C small c. This will be comparatively lighter than the darker skin. So you are seeing different phenotypes. Phenotypes as in physical appearance. So you see inheritance of skin color in humans show different phenotypes because different gene combinations are coming. Different genes are responsible for one particular trait. Different genes are responsible for one particular trait. So when different combination comes, you will find different colors as well or different phenotypes as well. So now next situation can be small a, small b, small c. When they come together, you get this combination, my dear children. Okay, now what do you call this? This is the lightest form that you can get, lightest skin tone that you can get. Okay, and what is this whole thing known as? This is known as polygeny, where multiple genes are responsible for one particular trait. Multiple genes are responsible for one particular trait, you call it polygeny. Works? Now, accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why? Because you lack, because there is a mutation in a gene which codes for phenylalanine hydroxylase, which is why all the phenylalanine is converted to phenylpyruvic acid and is stored there. If you want to read the answers, you can read like this. Okay, we will read skin color. It shows different phenotypes due to polygenic inheritance where multiple genes contributed to the phenotype, leading to a range of skin color variation. So you have multiple gene combination, so which is why you have multiple skin coloration as well. Works? Shall we go to the next question? Let's see. 23rd question. Define aneuploidy. Give 2 examples of aneuploidy. Okay. Aneuploidy basically means gain or loss of chromosomes. Gain or loss of chromosome is known as aneuploidy. Okay, 2 examples of aneuploidy. Turner syndrome, and then you want Down syndrome. Down syndrome. In Down syndrome, you say there is a gain of a chromosome. In Turner syndrome, there is a loss of a chromosome. Here it's a loss of a sex chromosome. Here is a gain of an extra autosomal chromosome. Okay. Now mention the karyotype of Klinefelter syndrome of an affected individual and write its symptoms. So what is the karyotype? As in, they are asking about the chromosome number and everything. So it's Klinefelter, so that means it is XXY condition. But how many autosomes do they have, children? They have 44 autosomes, and then 1, 2, 3. 44 plus 3, 47 total chromosomes are there. Okay, but they will be having this condition. Okay, and the symptoms we are going to see that in the next session. So let's see. First thing, we will see aneuploidy answer, and then we will talk about this thing, karyotype. So aneuploidy is a condition in which an organism has an abnormal number of chromosomes, either due to gain or loss of one or more chromosomes. It is caused by non-disjunction during cell division. Non-disjunction as in usually the chromosomes have to separate themselves during cell division. So just think that, sorry, this is a chromosome, and basically there is one more chromosome, okay? They have to separate, but these two guys are coming this side only. And now it comes this side, all the chromosomes are up here, this cell is divided, this cell is devoid of anything. You get it? So you are losing chromosomes over here in that particular case. That is what you mean by non-disjunction. They are not properly getting separated. Please remember this. So aneuploidy is what is a condition where there is an abnormal combination of abnormal number of chromosomes. Why? Because of gain or loss of the chromosomes. It is caused by non-disjunction of the chromosomes during the cell division. Example, Down syndrome where trisomy of chromosome number 21 is there. Turner syndrome where monosomy of X chromosome is there. 45 chromosomes will be there at the end. Now talking about Klinefelter syndrome, the karyotype of an individual affected by Klinefelter syndrome is 47 XXY. An extra X chromosome is there. So you can write like this, or you can better probably write like this and say 47 chromosome in total. Either way works. Symptoms of Klinefelter syndrome: underdeveloped secondary sexual characters. So just like how male will be having certain secondary sexual characters, for example growth of hair and things like that, they will be having it underdeveloped. Okay, gynecomastia, that is enlargement of breast in the case of males. Infertility due to lack of spermatogenesis. Tall stature with long arms and legs. Learning difficulty in some cases, but that is not very common. Okay, so we will restrict ourselves to these 4 things. Let's forget this. So underdeveloped secondary sexual characters, gynecomastia, and then infertility due to lack of spermatogenesis, sperms are not being formed very efficiently. Tall stature with long arms and legs. Works? That is what you mean by that. Now aneuploidy, everything results in chromosomal disorders. Okay, there is something that is known as polyploidy. It's there in your NCERT. Polyploidy means the whole set of chromosome is getting doubled or probably reduced. It happens in plants generally. That is also going to result in what? Chromosomal disorder. So aneuploidy, everything is resulting in what? Children, chromosomal disorders itself. Works? Okay. Next question. What is incomplete dominance? Explain it with reference to flower color in Snapdragon. So let's study incomplete dominance. It's very interesting. So suppose you have capital R, and you have smaller. This capital R basically refers to the color red. Okay, small r refers to the color white. We are talking about the flowers of Snapdragon plant. When capital R there, then probably it is red in color. Smaller, then it has to be what? White in color, right? This is parent. Now the gametes that will be produced. So it can be capital R, or it can be another capital R. Here it can be small r, or it can be small r. Agree? Now the F1 progeny that will be produced. This and this fuses to give capital R, small r. This and this fuses to give capital R, small r. This and this fuses to give again capital R, small r. Again capital R, small r, my dear children. All of you, if you see properly, there is capital R and there is small r. Okay, so usually when this was the condition, capital small t was there, this was overshadowing the effect and it was tall, right? But over here, there is a twist. This capital R is going to overshadow the effect over here, similarly over here, similarly over here, similarly over here. but not completely. But not completely, which is why all of them, they will be pink in color. So instead of being red, they are pink in color. Why, children? Because they are not completely dominant. This is known as incomplete dominance. It is known as incomplete dominance. Okay, that's not all. Listen to me. This is F1 progeny where all of them are similar, right? Now let's do self-crossing. Okay, let's cross both of them. So for that, again, we have to make parents. Okay, in parents you have capital R, small r, capital R, small r. The gametes will be capital R, small r, capital R, small r. Okay, when you fuse, what do you get? Let's see. When you fuse this and this, you get capital R, capital R. When you fuse this and this, you get capital R, small r, right? When you fuse this and this, you get capital R, small r. When you fuse this and this, you get small r, small r. Now if you see, this is capital R, this becomes red. This is capital R, small r, this becomes pink. And here they become white. So what is the ratio? Phenotypic ratio, how do they look? One is red, two are pink, and one is white. So phenotypic ratio, pheno as in physical appearance. Okay, how do they look? Phenotypic ratio will be 1:2:1. Okay, how will I write it? Red is to pink is to white. What is the genotypic ratio, my dear children? Genotypically, okay, genetic makeup, how it is? One is capital R, only one. Two of them are capital R, small r, two. And then one is small r, small r. So, genotypic ratio is what, children? Capital R, I'll write 1:2:1. So, both phenotypic and genotypic ratio is the same. 1:2:1. So, capital R, small r, and small r, small r. Works? Cool? So this is how you have to answer. So this is, you have to make this whole thing, and then you have to explain what is incomplete dominance. How do you explain? That basically in a heterogamous condition, one allele is not completely dominant over the another allele, which is why it is known as incomplete dominance. It will show an intermediate character. Okay? So, a phenomenon where both the alleles of a character express incompletely, producing a new intermediate phenotype. A new phenotype is being observed, that is pink color. In a heterozygous condition, please not forget to write this. Heterozygous, when this is the condition, that's when you find it, right? So, this is not same. Capital R is not there, capital R, small r is there. So, it is heterozygous condition. So, this is known as incomplete dominance. Now, Coren was a person who was behind it, okay? He crossed homozygous red flower, you are explaining, with homozygous white flower, and in F1 generation all hybrids were pink color. Okay, and you write the thing also, capital R, small r. Now the dominant allele R was not completely dominant over the recessive allele small r, and this made it possible to distinguish capital R, small r as pink from capital R, red, and small r, white. When F1 pink flowering plants were self-crossed, the F2 generation produced 25% red flower, 50% pink flower, and 25% white flower, as you can see over here. 25% red, 50% pink, and rest 25% white. From the hybridization result, we have observed that F2 phenotypic and genotypic ratio is this. So, will you be able to do that, children? You have to write in points and make them understand this. So, instead of making this, you can do the Punnett square also. Okay, either way. But please mention parent, gamete, F1 progeny. When you are writing answer, please mention heterozygous condition. Please mention, don't write 50% of this is what. You have to write that this is the condition, capital R, small r, or this is what, pink in color, whatever it is required, write it there. Okay, this is a normal picture of it for you to refer. Works? This came in 2023 March. Last question that we are having is, write the schematic representation of one gene inheritance in Pisum sativum. What is Pisum sativum? The garden pea plant. Okay, the garden pea plant. Now schematic representation, as in you have to write the whole thing and you have to explain. How will you explain? One gene inheritance. You're talking about just one gene. For example, gene for height. For example, what gene are you considering, children? This gene is for height. Okay. Now the alleles that you are having, one is tall, you write tall, small t is for dwarf. Okay. Then you explain that you are taking parents. What parents? Pure breeding parents. Pure breeding parents as in homozygous parents. Okay, they are homozygous, capital T and small t, small t. Then you are going to write gametes. What are the gametes? Capital T, small t and small t. Okay. Now what you find after parents, after gametes, what do you have, children? There is F1 progeny. How will you do? You have to fuse this with this. So capital T, small t. Then again capital T, small t. Capital T, small t. Capital T, small t. So all of them are capital T, small t that you are having over here. So basically that means all of them are phenotypic. Phenotype is what, children? Phenotype is tall. And what is the genotype of all of them, children? All of them, they are capital T, small t. Works? Write properly. Now next you are going to do self-crossing. So you make two of them like this and show that you are doing what? Selfing or self-crossing. Where this is the parent and now the gametes. For gametes you have capital T, small t, capital T and small t. Fuse them. Then what do you get? This and this. When you fuse you get capital T. This and this, when you fuse you get capital T, small t. This and this, when you fuse you get capital T, small t. Always write the dominant one first, okay? This and this, when you fuse you get small t, small t. So what is the ending of it? If you see 1 is to 2 is to 1. What is this ratio, children? Genotypic or phenotypic? This is what? Genotypic or phenotypic ratio? I am talking about genotypic ratio. There is one like this, two of them like this, one of them like this. So what is the genotypic ratio? 1 is to 2 is to 1. Now what is 1? What is 2? What is 1? Again, so you have to write it. Capital T, capital T, capital T, small t, small t, small t. This is what I am denoting with this. Okay. Next I will talk about phenotypic ratio. Phenotypic ratio. How do they look? So when I see one is tall, this one is also tall, this one is also tall, because they show complete dominance, not incomplete dominance. This is dwarf. So 3 of them are tall, 1 is dwarf. So I will write 3 is to 1. And then I will write, what do you understand by 3? 3 is what? Tall. So tall is to dwarf, my dear little children. Works? So this is how you have to answer. So this is a schematic representation of one gene inheritance in garden pea or Pisum sativum, my dear children. This came in May 2023. So you see, these type of questions are being repeated again and again, so you have to practice it. Incomplete dominance, this, that. So it's all important. And in incomplete dominance and this, you saw the difference. There the genotypic and phenotypic ratio was 1 is to 2 is to 1. Okay, over here the phenotypic ratio is different. Okay, 3 is to 1 is the phenotypic ratio. Please be aware of that. So with this, we have completed the topics, and I'll tell you, according to what I've understood, what are the important topics: chromosomal disorders, all the names, how they are caused. Just the last 2 pages of your NCERT for this chapter, very much important. You have to know what is a disorder, what is the chromosome number, which one is affected, autosomal or allosomal, what is it? You have to know it. Make a chart and keep it somewhere in your study place. Okay, put it somewhere. It is easy. You understood the concept, then it becomes very, very easy for you to understand things. So all the chromosomal disorder. Now sex determination in all type of organisms. Okay, what type of organisms, my dear children? You have different category. You spoke about mammals, that is birds and mammals as in human beings, and Drosophila also comes under this. Okay, mammals, Drosophila, they come under this. Instead of mammal, I'll write human because that's there in your book. Human. And Drosophila was one category. Then insect is another category. Then birds is another category. Then your honeybee, haplodiploidy thing, no, is another category. So you should be knowing about this. Which one of them show male heterogamity or female heterogamity? Things you should be knowing about that. Okay. Examples of pleiotropy and polygyny. There are so many questions that have come around phenylketonuria. Ketonuria, which is a chromosomal disorder, but is an example of pleiotropy. Skin coloration, which is an example of polygyny. The symptoms of this: inheritance of one gene that we just studied as of now. Okay, also incomplete dominance. Incomplete dominance, my dear children. Okay, and then you have certain other topics also, separate topics. Okay, linkage and recombination, multiple allelism, multiple alleles as in A, B, O, so multiple allelism, very important. So go through these topics again and again. Down syndrome is a very important disease over here in chromosomal disorder. Okay, Klinefelter's, Down syndrome, Turner's, all of them. Very important. Works? And also Mendelian disorders, please learn that also. I am not telling the entire chapter, but yes, the questions have come from this and I have covered almost all questions, right? You saw it. Okay, I took extra questions also to cover some things. So I hope it's clear to you, and if you still have any doubts, do let me know. But expect 2 5-marker questions coming from this chapter, 1 2-marker question coming from this chapter, and 1 MCQ coming from this chapter. Okay, so I hope you are going to score 13 on 13 in this particular chapter, my dear kids. And if you have any doubt, please feel free to question me, to ask me. I'm there to help you out. And if you liked or loved this particular session, definitely let me know, because it means a lot to me. I have put all of my effort over here. Yes, and I wish you all the best. Please do not be worried. We are in this journey. We are going to ace this. We are going to score really good marks, and that's my surety. But you just have to try hard. And if you are trying hard, my dear children, there is nothing that is going to stop you. And if you are liking this particular channel also, do not forget to subscribe to our cute little channel, that is Diksha Karnataka. So bye-bye. And one more thing, that is, if you want the notes for this particular chapter, for the questions and everything, you have to fill the form for the WhatsApp, okay, and the notes will be sent to you in your WhatsApp, so you don't have to be worried, but where will I find the notes? Works? Okay, so bye-bye, take care.